Okay students, in this particular video we are going to start with the next part of the chapter, that is molecular basis of inheritance. So in this particular chapter let us start with the search for genetic material. So in the previous video we have studied the structure of DNA as well as the structure of polynucleotide chain and the packaging of DNA, right. So coming to the next part of the chapter, that is search for genetic material. So in the earliest days when the scientists were unaware of the genetic material, okay, they were actually started or they actually began their study and their search for genetic material, okay. When the search for genetic material was began, okay, many of the scientists, different scientists have given their own ideas about what? The genetic material, okay, the molecule, that is genetic material. And the first and foremost idea was given by what? Griffith, okay, and he experimented upon the particular disease causing bacterium called as Streptococcus pneumoniae. Okay, Streptococcus pneumoniae. When he was working upon this particular bacterium, okay, when he was working upon this particular bacterium, he observed something and he has given the first idea of what? The DNA, okay. That is why this particular experiment was will come for at first when we go for search for the genetic material. So let us start with the Griffith's transforming principle, okay, Griffith's transforming principle, okay. So do remember that the Griffith, okay, in his time he was working upon the bacterium called as Streptococcus pneumoniae, as I have told you. When he was working upon the bacterium Streptococcus pneumoniae, he used to culture the Streptococcus pneumoniae in his what? Laboratory, in the petri dishes, okay, in the petri dishes, okay, the top view I will draw here, see. He used to culture the Streptococcus pneumoniae bacterium, and when he was culturing the Streptococcus pneumoniae bacterium, he found that, okay, he found that there were two different types of colonies, okay, he found there were two different types of colonies, okay. Those colonies are nothing but what? Okay, different in nature. That means some were smooth in nature and some were having rough surface, okay. Some were smooth in nature, okay. I'll just show this as smooth, okay. Some colonies were having smooth and shiny surface. Do remember, when he was culturing the Streptococcus pneumoniae bacterium in his laboratory, what he observed? In the petri dish, he observed two different types of colonies, okay, bacterial colonies. Out of those two different types of bacterial colonies, some were smooth in nature and some were rough in nature, okay. The smooth colonies were shiny in nature, okay, smooth and shiny, but rough colonies were not shiny. Is that clear? Then he got very curiosity about these particular smooth and rough colonies. Why the colonies are different even the bacteria is same, okay. Even though the bacteria, that is Streptococcus pneumoniae, is same, they found or he found two colonies, or they have formed two different colonies. Even though the bacteria is same, why and how they can form two different colonies, he got the curiosity. Then he checked, okay, he studied them properly and he observed them properly and came to know that the smooth colonied bacteria were having, okay, the smooth colonied bacteria were having a polysaccharide coat on them, okay, polysaccharide coat on them. But the bacteria belonging to rough colony, they were not having any polysaccharide coat, okay. Due to presence of polysaccharide coat, they were smooth and shiny in nature, okay. Due to presence of polysaccharide coat, they were smooth and shiny in nature. But, okay, in the other one, there was no presence of polysaccharide coat. That is why it appeared rough in nature. Okay, that is why it appeared rough in nature. Since he found smooth and the rough bacterium, he named them as what? The smooth colonied bacteria, he named it as S strain bacteria. Okay, he named it as what? S strain bacteria. And the another one, okay, where the rough colonies were formed, the bacteria which belongs to rough colonies, he named it as R strain bacteria. Okay, he named them as what? R strain bacteria. Very simple point. You know, he was culturing the Streptococcus pneumoniae bacteria, and in the petri dish he observed two colonies, two different types of colonies. Some colonies were smooth and shiny and other colonies were rough. Okay, he identified or he observed both of the bacterium from two different colonies separately and found that the bacteria belonging to smooth and shiny colony were having polysaccharide coat on them, but the rough colony bacteria did not have any polysaccharide coat on them. Due to presence of polysaccharide coat, they were appearing shiny, and due to absence of polysaccharide coat, the rough colony bacteria were appearing non-shiny. Is that clear? And ultimately what has happened? Okay, he named them as, okay, the one with polysaccharide coat, he named it as what? S strain bacteria. Okay, and the other one he named it as R strain bacteria. Okay, R strain bacteria. Is that clear? He did not quit it there itself. Okay, he did not sat quite there. Okay, what he did? He was curious about these two S and R strain bacteria, and what he did is he checked their virulency. Okay, first what he did in the first step, okay, I'll write it here, just see. Okay, in the first step what he did, he has taken the S strain bacteria and checked its virulency. How can he check the virulency? He simply just injected them into the mice. Okay, S strain bacteria he has taken and he just injected into the mice, as I have shown here. Okay, the mice were found to be dead. Okay, when he injected S strain bacteria into the mice, okay, when he injected S strain bacteria into the mice, okay, the mice were found to be dead. Why it happened? Because the S strain bacteria have caused the disease inside the mice. Okay, which disease? By the name itself you can understand, it is pneumonia. By means of pneumonia the mice died. Then he came to the conclusion that S strained bacteria is virulent. Okay, he came to the conclusion that S strained bacteria is virulent. Okay, then he checked the same for R strain. He has taken R strain bacteria, then he introduced the R strain bacteria into the mice. Okay, then what has happened? The mice did not die, they were left alive. Okay, they were left alive. When all the mice were left alive, even though injecting R strain bacteria, then he concluded that R strained bacteria, okay, R strained bacteria is non-virulent. Okay, non-virulent. Is that clear? So the same is given in this diagram. You can see that R strained bacteria, okay, when he injected the same inside the mice did not die, it was left alive. Okay, but S strained bacteria or smooth strained bacteria, when the smooth strained bacteria are injected into the mice, they died because the S strain was what? Virulent in nature. Clear? He did not sit quite up to this one, okay, or at this particular point he did not sit quite. What he did? See, further what he did is, in the third step he heat killed. Okay, what he did? he heat killed the S strain bacteria. Okay, S strain bacteria was what? Actually virulent form, and this virulent form, what he did? He heat killed them. Okay, by means of giving the heat treatment, he killed all the S strain bacteria, then injected into the mice. Okay, at that time he found that all the mice were left alive. That means no mice died, because he has done heat killing of what? The S strain bacteria. So it clearly tells that heat killed S strain bacteria is what? Non-virulent. Okay, heat killed S strain bacteria is non-virulent again, right? Then further he has taken the next step. Okay, what he done is, he taken heat killed S strain bacteria and mixed them with what? Live R strain bacteria. Okay, he mixed them with what? Live R strain bacteria, and he then injected the combination of both of these to the mice. Okay, as a result of which, what has happened? The mice were, okay, found to be dead. Okay, but do remember, we know that heat killed S strain bacteria is non-virulent, live R strain is also non-virulent. When we combine both of them, okay, both are non-virulent forms, that means the mice should not die. Is that right? But surprisingly, what has happened? The mice dies here. Okay, surprisingly, what has happened? When, even though both of them are non-virulent, when the mixture of both of them is given to the mice, surprisingly all the mice were found to be dead. Okay, the reason behind this one, he was searching. Okay, and what he did was, he has taken the dead mice and isolated, okay, the bacteria from the dead mice. When he isolated the bacteria from the dead mice, he found that the presence of live S strained bacteria in them. Okay, when he injected the heat killed S strain and live R strain bacteria into the mice should not supposed to be dead, but here the mice died. Okay, then he got confused why the mice died, and he was about to search it. That is why he took the dead mice and isolated the bacteria from it. When he isolated, he found that there were presence of live S strain bacteria, but in the injected bacteria, he has injected heat killed S strain as well as live R strain, but whatever are collected from the dead mice, he found totally different type. That means live S strain bacteria he found. Then he got the question. Okay, the possibilities are of two types here. One, the heat killed S strain bacteria have become alive again. So I will write the possibilities here, first one and second one. First possibility is heat killed, okay, S strain bacteria have become alive again, or second possibility is the heat killed S strain bacteria have made the live R strain, okay, to get converted into live S strain. Okay, these were the two thoughts that strike in his mind. But in the first one, see here, in the first one, heat killed S strain, whether will they become alive again? No, not at all possible, because the dead organisms cannot become alive again. That is why this possibility went wrong. Okay, but second possibility is there. That means heat killed S strain bacteria have given something to the live R strain bacteria and made them to undergo the transformation into what? Live S strain. This transformation will be taking place anta Griffith helda. Okay, the Griffith said that, okay, the Griffith concluded that what has happened? The heat killed S strain bacteria have transformed live R strain into live S strain. Okay, and this transformation of live R strain into live S strain is due to the transforming principle that is present with the what? Heat killed S strain bacteria. Is that clear? That was explained by Griffith. Okay, but he did not say which molecule is transforming the live R strain into live S strain. What he said, some transforming principle is there with the heat killed S strain bacteria that is transforming or that is changing the live R strain into live S strain. Clear? And this itself is called as Griffith's transforming principle. Okay, according to Griffith's transforming principle, what will happen? The heat killed S strain bacteria, okay, have somehow transformed live R strain bacteria into live S strain. Okay, and some transforming principle is working here where it is making the live R strain to get transformed into live S strain, and this was explained by the Griffith, but he did not specify any of the molecule here that will help for the transformation. Is that clear? He finally told that some transforming principle is working, that is it. And further, the biochemical characterization was done by, okay, biochemical characterization of what? Griffith's experiment. So, what do you mean by biochemical characterization of Griffith's experiment? Very simple. It is nothing but the experiment of Griffith, which was concluded by Griffith, was continued by what? By the three scientists, namely Ostwald Avery, Colin MacLeod and Maclean McCarthy. So, these three scientists in the year 1933 to 1944 have continued the Griffith's experiment and found that which molecule is transforming the live R strain bacteria into live S strain. First of all, what they have done, I will explain you here. Okay, they have taken heat killed S strain bacteria. Okay, they have taken heat killed S strain bacteria, and what they have done, they have isolated the major molecules which are present in it. Okay, major biomolecules which are present in heat killed S strain bacteria were isolated first, and they found that there were DNA and the RNA and the protein. So, these three were the major molecules which were present in what? The heat killed S strain bacteria. Clear? And out of these three, okay, out of these three, one will be helping for transformation of the heat killed transformation of R strained bacteria into S strain bacteria. Okay, and for that, what they have done? Okay, they have done one particular point. They have used some of the enzymes to destroy them one by one, and they have added one by one to the live R strain. Okay, as explained now. Okay, I will explain it now. See what they have done. They have taken heat killed S strain bacteria. To this, they have added what? The enzyme DNase and the enzyme RNase. Okay, when to the heat killed S strain bacteria DNase and RNase enzymes are added, the DNA and RNA of heat killed S strain bacteria will be destroyed, because these are the lytic enzymes. The DNase enzyme will destroy the DNA and RNase enzyme will destroy the RNA. Ultimately, what will be remaining? The protein part will be left as it is. Then it is mixed with live R strain bacteria, then injected into the mice, and the mice were not died, or the mice were left alive. It means that protein is not converting or not transforming what? This particular live R strain to live S strain. So, in the next step they have changed it. they have taken heat killed S strain bacteria to which they have added DNase and protease. Okay, that means they have destroyed DNA and protein. Okay, they have left only the RNA of heat killed S strain bacteria to get mixed with what? Live R strain bacteria. When they have done that, okay, and allowed or injected it to the mice, again the mice were found to be alive. That means the mice did not die, right? That means even the RNA is also not responsible for what? The transformation. Finally, they have checked it for the DNA. They have taken heat killed S strain bacteria to which they have added RNase and protease. Okay. When they have added RNase and protease to the heat killed S strain bacteria, RNA and proteins were destroyed from the heat killed S strain. Then what is left? Only the DNA is left, and that DNA was mixed with live R strain bacteria, then injected it into the mice. When they have injected it into the mice, they found that all the mice were dead. Is that clear? So, by this particular step, it is clear that DNA is transforming. What? The DNA is transforming live R strain, okay, live R strain bacteria into live S strain bacteria. Is that clear? Live R strain bacteria into live S strain bacteria. Okay. This exactly was concluded by the biochemical characterization of Griffith's experiment by these three scientists. Okay. Thank you. Hello students. Okay, let us continue with the next part of the chapter, that is Hershey's and Chase experiment. Okay, Hershey's and Chase experiment. So, very important point to note down here is the Hershey's and Chase experiment is the first experiment to prove that DNA itself is the genetic material. See here, Hershey and Chase experiment itself is the first experiment to prove DNA is the genetic material. Okay. So, what exactly Hershey and Chase did? Okay. Let us start with this particular experiment. Okay. The scientists Hershey and Chase, they have observed what? One particular phenomenon of what? The infection of, okay, bacteriophage in a particular bacterium. This is a bacterium. Okay, this is a bacterium, and this bacterium was attacked by what? The bacteriophage. Okay, was attacked by bacteriophage, which was what? A virus. The virus that attacks the bacteriophage, okay, is called as, the virus that attacks bacterium is called as bacteriophage. Okay, the virus particle that attacks a bacterium is called as bacteriophage, and the attack of bacteriophage on a particular bacterium was studied first properly by Hershey and Chase. And during this particular study, what they have observed is, they have observed the virus particle cannot enter into the cell completely, or the virus particle will not enter into the cell completely to cause the infection. Okay. Instead of entering the cell completely, what it will do? It will just send its genetic material inside the cell. Okay. The virus particles will not enter into the cell completely. What they will do? they will just send their genetic content into the bacterium or into the host cell for the further infection. So, this clearly tells that what? The material that enters into the host cell will be the genetic material of that particular organism. Is that clear? And the virus, if it injects, okay, the virus if it injects the protein coat, okay, if it injects the protein, then protein will be the genetic material. If it injects the DNA, then DNA will be what? Genetic material. Is that clear? So, what the Hershey and Chase did, they have identified which particular molecule can enter inside. Okay. For that sake, they have labeled, okay, the particular molecules which are present in this particular bacteriophage. Right. Let us study about that in detail. Okay. See, Hershey and Chase, what they have taken? They have taken some bacteriophages, okay, bacteriophages, okay, and they have developed the bacteriophages in two separate containers, okay, in two separate containers. In one container or in one particular petri dish, the medium was containing what? Radioactive phosphorus. Do you remember? In the first one, it was containing radioactive phosphorus. Let us consider this setup as A. Okay. The medium was containing what? Radioactive phosphorus. Do you remember? Radioactive phosphorus. And in the second one, okay, in the second medium, they have grown, okay, the bacteriophages, and this medium was containing radioactive sulfur. Okay. The first one, okay, the setup A, this was containing what? The medium having radioactive phosphorus, and the setup B, okay, in this the medium in which bacteriophage were grown was containing radioactive sulfur. And for several generations, the bacteriophages are cultured in these two media. Okay. Later, what has happened? The bacteriophages which are obtained, okay, the bacteriophages which are obtained after several generations, okay, from these two media, they have given or they have shown the radioactivity. Okay. In the first one, okay, in the first one, the radioactivity was found in their what? DNA. Okay. Because phosphate group is present inside the DNA. Is that clear? The phosphate group is present inside the DNA. That is why the radioactivity, okay, was found in the DNA. See here, I will show it with a different color. Okay. This blue color indicates radioactive material. That is it. Okay. So, when the radioactivity was found in the, see here, okay, this is DNA, okay, that is radioactive in nature. Radioactive DNA. Is that clear? Radioactive DNA. Why? Because in the first setup, the medium was containing radioactive phosphorus. Okay. Since the medium was containing radioactive phosphorus, they have developed their DNA as radioactive molecule. Why? Because the DNA contains the DNA contains what? Phosphate group, and this phosphate group, since it is radioactive in the medium, the newly formed DNA became what? Radioactive in nature. Okay. In the second setup, what has happened? Okay. In the second setup, no doubt the bacteriophages, okay, in the second setup, the bacteriophages have got the radioactive protein coat. Instead of having the DNA, here the protein coat has become what? Radioactive. Why the protein coat has become radioactive here? Because what they have given, okay, they have given the radioactive sulfur in the medium. If the sulfur is radioactive, the radioactive sulfur will be entering into the protein, because the proteins are made up of what? Sulfur, or the proteins contain sulfur. That is why the radioactivity was found in the what? Outer protein coat. Okay. and inside the DNA was left normal as it is. Is that clear? That means what they have done, simply they have labeled the protein coat and the DNA with the help of radioactive phosphorus and radioactive sulfur. Okay, with the help of radioactive phosphorus they have labeled the DNA, and with the help of radioactive sulfur they have labeled the protein coat. Then what they have done, they allowed these particular bacteriophages, okay, to attack upon what? The particular bacterial cells. Okay, the bacteriophages were left to attack upon what? The bacterial cells. When they are left to attack on the bacterial cells, as usual they have inserted, okay, they have inserted their genetic material inside this one. See here, okay, they have injected their genetic material inside the bacterial cell. Okay, as usual they will inject their genetic material. We don't know which one it is. Okay, they will inject it, and the injected material will stay inside the cell, and after, okay, proper blending, okay, after proper blending or else vigorous mixing, what will happen? The outer side, whatever it is there, okay, it will be separated. Andre, soon after infection what they will do? They will inject their genetic material into the cell. When they inject their genetic material into the cell, the genetic material enters inside the cell and remaining part will be left outside. Is that clear? And after vigorous shaking or blending, what will happen? The material which is present outside, it will be separated. Okay, then what they have done? They have taken both of them into two separate test tubes, okay, and made to undergo centrifugation. Okay, when they made to undergo centrifugation, okay, when they have made to undergo centrifugation, what has happened? In the first one, the radioactivity was found at the bottom. Okay, in the first setup, radioactivity was found at the bottom. In the second setup, radioactivity was found at the supernatant. It means that why the radioactivity was found at the bottom here in the first setup? Because the bacterial cells are heavier and the particle has entered inside, it has settled at the bottom by means of what? Centrifugation. Centrifugation ಆಗಿದ್ದಕ್ಕೆ ಏನಾಯಿತು? Heavier particles have settled. Heavier particles ಅಂತಂದ್ರೆ nothing but the bacterial cells, they will be heavier. Okay, inside the bacterial cell what is there? Radioactive DNA is there, that is why radioactivity was found at the bottom. And in the other setup where we have labeled the protein coat, there the radioactivity was found at the supernatant. That means the protein coat has not entered inside the cell, instead the DNA has entered inside the cell. Since the DNA has entered inside the cell, the DNA will be what? Proved as genetic material. Is that clear? ಯಾವುದು ಒಳಗಡೆ ಹೋಗೈತಿ? DNA ಒಳಗಡೆ ಹೋಗೈತಿ, ಸೆಲ್ ಒಳಗಡೆ. ಹೌದಾ? DNA ಸೆಲ್ ಒಳಗಡೆ ಹೋಗಿದೆ ಅಂತ ಆದರೆ ಏನಾಗುತ್ತೆ? DNA will be the genetic material. Is that clear? DNA will be the genetic material and the other one will not be. That means protein coat is not the genetic material, DNA is the genetic material. So this was proved by what? Hershey and Chase experiment. Very important point here. Okay, you have to remember, very important point. DNA is the genetic material. Okay, DNA itself is the genetic material ಅಂತ ಪ್ರೂವ್ ಆಗಿದ್ದು ಅಥವಾ ಪ್ರೂವ್ ಮಾಡಿದವರು ಯಾರು? Hershey and Chase. For the first time it was proved by Hershey and Chase, that is DNA itself is the genetic material. Right, coming to the next part of the chapter, that is properties of genetic material. Okay, properties of genetic material. So now you came to know that genetic material itself, what? DNA. Okay, DNA itself is the genetic material. So if DNA is the genetic material, then what are the characters of a model genetic material? Okay, what are the characters? Okay, what are the characters of what? Genetic material. So first property of genetic material is it should be able to generate its replica. That is, it should undergo self replication. Okay, it should undergo what? Self replication. Okay, it should undergo self replication. Sorry, I have made a mistake here with the, okay, It is self-replication. Okay, it should undergo what? Self-replication. First point, if you want to call any molecule as genetic material, it should undergo self-replication. Okay, it should undergo what? Self-replication. That is the first point. That means it should be able to, or it should be capable of producing its own copy. Okay, it should be able to produce its own copy. That is the first property of genetic material. Then what about the second one? Okay, the second one is it should be stable chemically and structurally. Okay, that means the genetic material, whether it may be any of the molecule, it should be what? Chemically and structurally stable. That means it should not be, okay, it should not be reactive. Okay, it should not be reactive. Is that clear? That means whatever genetic material you take, for example, it should not be reactive in nature. Okay, it should not readily react with any of the molecules that are present, right? It should be stable. Okay, it should not mingle with any of the chemical reaction. Is that clear? Depending upon the temperature or depending upon any of the external factors, okay, they should not. That means the particular genetic material should not show any of the changes. It should not undergo any changes. Is that clear? Then next one. Next one is the third property of genetic material. It should provide the scope for slow changes, okay, or mutations that will be required for what? The purpose of evolution. Okay, slow changes should be seen, or slow changes should be occurring in the DNA or occurring in the genetic material. Whatever genetic material, whatever molecule you take it as genetic material, it should not be reactive, but it should show slow changes. Okay, so which will lead to what? The mutations. Okay, by means of these mutations, what will happen? Okay, it will lead to the evolution. Okay, it will lead to what? The evolution. Clear? For the purpose of evolution, it should show some slow changes that are called as mutations. Clear? The next one, fourth point. Okay, it should be able to express itself in the form of what? Mendelian characters. It should be able to express itself in the form of what? Mendelian characters. Okay, ಅದೇನಾಗಬೇಕು ಅಂತಂದರೆ ಮೆಂಡೆಲಿಯನ್ ಕ್ಯಾರೆಕ್ಟರ್ಸಲ್ಲಿ ಎಕ್ಸ್ಪ್ರೆಸ್ ಆಗಬೇಕು. ಓಕೆ, ಇಟ್ ಶುಡ್ ಬಿ ಏಬಲ್ ಟು ಎಕ್ಸ್ಪ್ರೆಸ್ ಇಟ್ ಸೆಲ್ಫ್ ಇನ್ ವಾಟ್? ಮೆಂಡೆಲಿಯನ್ ಕ್ಯಾರೆಕ್ಟರ್ಸ್. ದಟ್ ಮೀನ್ಸ್ ಇಟ್ ಶುಡ್ ಗೆಟ್ segregated during gamete formation. Is that clear? It should show segregation during gamete formation and it should show union during zygote formation. Okay, gamete formation ಆಗಬೇಕಾದ್ರೆ ಸಪ್ರೇಟ್ ಆಗಬೇಕು, ಅದೇ zygote formation ಆಗಬೇಕಾದ್ರೆ ಯೂನಿಯನ್ ಆಗಬೇಕು. Okay, and independent agi separate agbeku, independent assortment and all those things. Okay, so these are what? The basic properties that a particular genetic material should have. Okay, basic properties. Gotaita? Then coming to the next point, that is, see, coming to the next point, that is DNA versus RNA, which I have mentioned here. Okay, DNA versus RNA. Okay, see, DNA versus RNA. What do you mean by DNA versus RNA? DNA or RNA, both are going to act as what? Genetic material, no doubt. Okay, both of these molecules, that is DNA and RNA, they will act as genetic material. Okay, no issues. But out of this DNA and RNA, which one is better genetic material? Okay, out of the DNA and RNA, which one is better genetic material? So if you ask this particular question, then the answer will be what? Okay, DNA is better genetic material when compared with the RNA. Okay, why DNA can be the better genetic material, okay, when compared to RNA? That I will express here. See here. Okay, so if you consider DNA and RNA the better genetic material will be DNA. Why? Because DNA is first of all, okay, double stranded. Okay, DNA is double stranded and RNA is single stranded. Okay, DNA is double stranded and RNA is single stranded. Is that clear? Due to the double stranded nature of the DNA, it will be more stable when compared with the single stranded RNA. Why? Because say, if you consider this as the DNA, okay, if this as the DNA, here all the nitrogenous bases are paired in nature. Okay, nitrogenous bases will be what? Paired in nature, that is why DNA will be stable. Okay, that is why DNA will be stable. But when you see the RNA, it will be having unpaired nitrogenous bases. When the unpaired nitrogenous bases are present, they want to get paired up always. That is why RNA will be not stable, it is reactive in nature. Okay. Second point, okay, DNA has got thymine. Okay, DNA has got what? Thymine as its pyrimidine base. But in case of RNA, instead of thymine, what is there? Okay, the uracil is there. Okay, presence of uracil will not give stability to the RNA. Okay, presence of uracil will not give stability to RNA. But in case of DNA, uracil is modified into what? The thymine. Okay, in case of RNA, in case of DNA, what will happen? The uracil will be modified. Okay, in the DNA, uracil will be modified to form the thymine that gives more stability. Is that clear? Uracil is not highly stable, or it is not stable when compared to thymine, but thymine is what? Stable molecule, right? Third point is, in case of DNA, OH group is absent. Okay, OH group is absent, but in case of what? The RNA, OH group is present. So presence of OH group again is a drawback for the RNA. Why? Because it will make it more what? It will make the particular RNA more reactive. It will make the RNA more reactive. Clear? So these are all the different points that makes DNA as a successful genetic material when compared with the RNA. Is that clear? So coming to the next one. See, let us go to the next one, that is RNA world. Okay, that is RNA world. What do you mean by RNA world here? Okay, RNA world is nothing but the first formed world, that means primitive world. In the beginning of life forms, okay, the beginning of life forms, when the organisms were formed, they all used to have RNA as their, okay, they all used to have RNA as their genetic material. Okay, earlier days, okay, all the organisms such as viruses, okay, and primitive organisms, bacteriophages, tobacco mosaic virus, etc., they used to have what? RNA as their genetic material. Okay, why they did have RNA as their genetic material? Okay, because they were primitive in its nature. Okay, primitive nature was there. Earlier world, most of the organisms were having RNA as their genetic material. But later on, what has happened? The organisms started to evolve. Okay, the organisms started to evolve, and they have developed the DNA as their genetic material. How the DNA has developed from RNA? Very simple. RNA is single stranded, and this single stranded RNA was found to be more reactive and unstable. Okay, they have developed another strand over this one, and the uracil is replaced with the help of thymine, so that they developed what? The DNA. Okay, so by means of developing the DNA, the organisms have got what? The more stable genetic material, that is DNA. Okay, firstly or at the beginning, the life forms used to have what? RNA as their genetic material. Most of the living organisms in the beginning of the what earth or beginning of the life, okay, they did have what? The RNA as their genetic material. That was called as what? RNA world. Okay, at that time maximum organisms used to have RNA as their genetic material. But later on, what has happened? RNA was developed or transformed into what? DNA. Okay, then what will happen? The most stable genetic material was formed, that is DNA. Right. Next one, coming to the next part of the chapter, very very important for your NEET as well as for your theory examination, that is, okay, very important for your NEET examination as well as what? The theory part. So, that is called as what? DNA replication. So, what exactly is meant by DNA replication? Okay, what is meant by what? DNA replication. Do remember, DNA replication is nothing but, okay, the process of, I will write it here, the process of formation or you can say this as the biosynthesis. Okay, process of biosynthesis or formation of DNA from parental DNA strands. Okay, daughter DNA from parental DNA. Okay, so that is called as what? The process of replication. Okay, replication antandre eno? The process of formation of daughter DNA from the parental DNA. Okay, that is called the DNA replication. Okay, see, replication of DNA is nothing but, it is copying of DNA from parental DNA. And Watson and Crick, okay, the scientist Watson and Crick, who had proposed the structure of DNA, themselves have given the idea of what? DNA replication. And they said that, okay, it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism of the genetic material. That means, when they were studying the structure and when they were proposing the structure of DNA, they said that, okay, they came to know that the replication, how it exactly happens, that was also came to their view. Okay, they also came to know that how exactly the replication happens, and they said that DNA replication is what? The DNA replication is semi-conservative. Okay, DNA replication is what? Semi-conservative. What do you mean by semi-conservative method of DNA replication? Here, what happens? If the parental DNA strands are like this, okay, if the parental DNA strands are there like this, then what will happen during the time of replication? Both of these parental DNA strands will get separated from each other. Okay, both of them will get separated from each other, and on these separated strands, what will happen? Okay, sorry. Both of them will get separated from each other, and when they get separated from each other, on these particular parental strand, new strands will be formed like this. Okay, new strands will be formed, which will give rise to two daughter DNA molecules. So, this process, okay, of formation of daughter DNA from the parental DNA is said to be semi-conservative. Why it is said to be semi-conservative? Just you can observe here, one strand is newly synthesized. Okay, here also only one strand is newly synthesized, but what about the other strands? The other strands are taken from the parent. That means 50% of the strands are newly synthesized and other 50% are what? The parental DNA. Is that clear? Since only 50% is newly synthesized, it is called as semi-conservative. That means the conservation of other 50% formation of the DNA has been taken place there. Is that clear? So, this particular process of semi-conservative DNA, okay, was told or was explained by whom? The particular scientist, that is Watson and Crick. Coming to the next one, okay. Coming to the next one, how exactly the replication process takes place. Okay. Next one is the process of DNA replication. How exactly the DNA replication takes place. Okay. Very simple. The process of DNA replication takes place by means of what? The group of enzymes. Okay. By means of group of enzymes. Right. See, what happens when the DNA replication starts, okay, the first point is at one point of the DNA. If you consider this as DNA, at one particular point of the DNA, what will happen? The strands will be separated by means of breaking down what? The hydrogen bonds. Okay, the hydrogen bonds will be present like this. So, this is DNA molecule and the hydrogen bonds will be present between the base pairs, right. What will happen during the process of replication? The first step that takes place, that is, okay, it starts at one particular point called as Ori site. Separation of, okay, separation of DNA strands will take place. Separation of DNA strands will take place at one particular place called as Ori site. You can call it as Ori site or site of origin of replication. Okay. Then a unit of replication with one origin is called as replicon. Do remember, separation of DNA strands will take place at one particular point. It is called as what? Ori site. Okay. Then the strands will be like this. Okay, the strands will become like this. Why? Because it has led to breaking of the bonds there in between, and ultimately they are separated. Okay. So, this will lead to what? Separation of both the strands here like this. Okay. The strands are separated. Then what will happen? Okay. I will just show you the polarity, 3 dash and 5 dash, here 5 dash and 3 dash, right. So, after formation of the Ori site, what will happen? The separation will continue. And do remember how exactly the separation of the strands will take place. The separation of the strands will take place by means of a particular enzyme called as DNA helicase. Okay. By the help of DNA helicase enzyme, both the strands will get separated to form the replication fork. So, this will be taking place. Here the separation has been taken place, and on both the direction, okay, the replication and separation will be initiated. Okay. And in both the directions similar processes will be taking place. Okay, similar processes will be taking place. That is why I will take only one part. Okay, only one part, and I will draw it as straight one here. You can come to know very easily. This is called replication fork. Okay, this is called replication fork. And here what has happened? By means of enzyme helicase. So, this is what? Enzyme helicase. With the help of enzyme helicase, which is moving in this direction, it is separating the DNA strands. Okay, here 3 prime end is there, here 5 prime end is there, and here 5 prime end will be there, here 3 prime end will be there, right. So, this enzyme helicase is moving in this particular direction and separating the strands, and here you can see the strands that are separated, and this particular structure with 2 separated strands, this will be called as what? Replication fork. You can see the replication fork, right. This is what? Replication fork, right. On these 2 strands which are separated here, okay, on these 2 strands, so this is strand 1 and this is strand 2. So, on these 2 strands which are separated, okay, you can see the unpaired nitrogenous bases, okay, unpaired nitrogenous bases, and these unpaired nitrogenous bases will be helping out, okay, to the formation of new strand over them. They will act as templates, okay, these strands will act as what? Template, okay. This is nothing but what? If on this particular template strand, if the unpaired nitrogenous base is A, then it will always attach with what? T. That means it will guide the formation of other strand here, okay. If on this strand, if it is A is there, and it will guide the T for the attachment here, and if it is G, and it will guide or it will attract the C. Like that, formation of new strand will be taking place. But one particular point is there, the formation of new strand will take place only with the help of what? Okay, the polymerase enzyme. You know that polymerase enzyme will help for what? Polymerization or formation of new DNA strands over the template strand, and the polymerase enzyme will be initiated or will start its function only if, okay, only if it gets a particular primer, okay, it gets a particular primer attached here. If the primer attachment has been taken place, RNA primer, do remember, okay, RNA primer, if it is attached, then what will happen? To that particular site, okay, to that particular site where the primer attachment has been taken place, there the polymerase enzyme will bind and start the process of what? Polymerization, okay. This is polymerase enzyme. Polymerase enzyme binds where? The region where what is there? Okay, primer is attached, okay, primer is attached, and do remember it is RNA primer, okay. The region where RNA primer is attached, at that particular point the polymerase enzyme will come and bind to initiate or to start what? The process of polymerization. When the process of polymerization is initiated, then what will happen? The formation of new strand will be taking place in this particular direction, okay, and these are the nitrogenous base pairing, okay, and formation of new strand will take place. Do remember that formation of new strand of the DNA will be always taking in the direction of 5' to 3', okay. Very important point it is. Formation of new strand of the DNA will be always taking place in the direction of 5' to 3'. So, if the template strand is having 3' here, then here the formation will be initiated at 5', isn't it? So that in this direction, if the formation of strand take place, it will be taking place continuously, right. But on the other hand, the other template strand has got what? 5' at this particular end. So the formation of the new strand of DNA cannot takes place in this direction, okay. So this direction, it is not possible for the formation of new strand. It should always take place in the direction of what? 5' to 3' only. Is that clear? For that sake, what will happen? It should wait up to the opening of what? This fork, okay. It should wait up to the opening of this fork, and here now it is open, okay, up to a certain extent. So what will happen? The primer will be attached here, and when the primer is attached here at this particular position, then the formation of the strand will begin and it will be continued up to this extent, right. Then second time at which, okay, I will show you. When the second time separation takes place, further separation of the coil, when it takes place, okay, like this, no doubt this particular strand which will be having 5' to 3' direction, it will continue, okay. But so this will be having only this much of strength and other thing will be initiated like this. Is that clear? Andre, as it go on, as the helicase enzyme go on separating the strands, then what will happen? The unpaired strand will be formed. Is that clear? On fragments of DNA will be formed, and these fragments of DNA which are formed on the strand which is having the polarity 5 prime to 3 prime called as what? Okazaki fragments. ಅವಕ್ಕೇನಂತ ಕರಿತೀವಿ? Okazaki fragments. Okay, Okazaki fragments they are called as, which were first identified by what? The scientist called as Okazaki. ಅದಕ್ಕೆ ಅವಕ್ಕೇನಂತ ಕರಿತೀವಿ? Okazaki fragments ಅಂತ ಕರಿತೀವಿ. And to remember, each and every Okazaki fragment will be having the RNA primer here. Is that clear? Then with the help of this Okazaki fragments, okay, the another strand will be formed. But what is the difference between both of these is, one strand is forming continuously without any break, but the other strand is having many breaks here. That is why one strand which is having the polarity, the template strand, do remember, I will write it here, very important point, do remember, I will write it here. The template strand, if it is having 3' to 5' polarity, on this template strand continuous synthesis will take place, okay, and it will lead to what? Leading strand. It is called as what? Leading strand, okay. And on other template strand which is having the polarity, that is 5' to 3', on this particular strand what will happen? The synthesis of new strand will be taking place by means of bit by bit, called as Okazaki fragments. That is why here the process of formation of new strand will be slow, hence it is called as lagging strand. ಅದಕ್ಕೆ ಏನಂತ ಕರಿತೀವಿ? Lagging strand ಅಂತ ಕರಿತೀವಿ, okay. On which the continuous synthesis is taking place, it is called a leading strand, and on which the continuous synthesis is not taking place, it is called as lagging strand. ಗೊತ್ತಾಯ್ತಾ? Leading strand and lagging strand. Ultimately what will happen? The lagging strand which has got Okazaki fragments, these fragments will be joined, okay, end to end with the help of what? The enzyme called as DNA ligase, okay. Do remember, the strands, okay, Okazaki fragments, okay, the Okazaki fragments or fragments of the DNA, they are joined with one another with the help of what?